Module 3, developing genetic tests. This really addresses the CLIA labs. What are they? How do they get set up? What are the tests that go into them? What's the difference if you're asked about a lab designed or developed test versus an FDA cleared test? I'm going to address that, bring some of this knowledge base to the table, just really have that information. You don't have to spout it off, but more really just understanding what your test is doing in that CLIA lab, and the fact that there is a lot of regulatory compliance, that there's a lot of quality assurance. These are the key words that you want to be throwing out there and talking to your customers so they get that this is not done in some shack in the back of someone's garden. This is an actual lab that has been accredited, it has been reviewed, audited, etc. So let me take you through some of these slides and then this will wrap up today. So this is a little picture I took from the FDA, and the FDA, something for you to be aware of is that the FDA's views on CLIA labs are a little bit different than those people that use CLIA labs, and I'll explain why. Now,
and you're saying clear at
CLIA.
C-L-I. I'll tell you what it means in a minute. It's up on the slides.
Thank you. I thought you were saying clear.
It's now clear, right? Clear.
Yes.
Clear to have clear.
So it's really about, you have gone in and asked your customers what is it you need, and then you have a design input, design output, and the processes in between. It's review steps all the way through. There's a verification step that verifies that the design inputs equal the design outputs, and then the medical device, which in this case is your lab design tests, which are all these panels, which are then used in the CLIA lab, are validated. Okay, so I believe Dr. Cohen has gone through all of this, and all the documentation there has been put into place. There are SOPs put in place. It's extremely rigorous. Okay, so bear that in mind. Rigorousness, documentation, traceability, quality. So when you are designing a molecular diagnostic assay, there are various phases that you have to go through. There is designing the PCR. This is molecular based, which means DNA. You can either create a singleplex or a multiplex. Now, we talked about the process behind these panels. This is a multiplex. So you then go through product development as you develop this assay and verify that it works over and over again, at least 3 times. And there are phase gates. And then you have to transfer this over to a manufacturer, which is not your case, but know this, an FDA cleared product has to go through these processes. So you will be asked, well, what's the difference between your test and this FDA cleared test? Okay? So there's a step in there where there's a manufacturing step where all this information that came out through product development is transferred over to the manufacturer, who then copies everything and builds the kit, and then it gets commercialized. There are 3 roads to IVD products. Very simply, when you have gone through your research phase and understood what it is you're building, you then go through the stage gate process, which is feasibility, verification and validation, and then you go to the manufacturing. Now, if you're not manufacturing, you go straight into this step here, which is a validation on what I say retrospective samples. You've got to test it out on something. So retrospective samples is always the place that these type of tests go into. FDA cleared tests have to go through a clinical study, which means prospective. There are patients that walk in, they have the cancer, the samples are taken, bang, it's tested. This then gets put together in a nice document submitted to the FDA, and they clear it. They say, this is a good test, looks like you've done everything well, we're giving you 510K. When you are creating a product in a CLIA lab, C-L-I-A, it's a lab designed test which doesn't have to go through a clinical trial, but does need to be tested on retrospective samples that are already out there. These FFP blocks, you can buy them, you can test them, that have been tested or not tested, but have been reviewed by a pathologist and assigned lung cancer. So you put your lung cancer panel on, then you test it. That's it. That's all you need to do. But that's what you need to do to get the LDT, the lab developed test, through into the CLIA with all the documentation in place. And then a final one which I put up here was the CE mark. It's somewhat similar to the clear lab design test, again, it's retrospective samples. And for Europe, they're very happy with this. They don't go into this incredible level that the FDA expects companies to go through to get the product into the US market. That's why clear labs have sprung up all over the place. That's why these lab design tests are put into the clear lab, because basically companies can't afford to follow the rules and regulations of the FDA. And so there's this big discussion about how much the FDA then should impose on clear labs to bring their level up to a higher level than what they're already at. And this is the big pinch between companies that are trying to make a business out of this, diagnostics. So designing is really about, you pick your targets of interest. In the NGS process, it's multiplexing. It's typically done with a software. You know, I mentioned down here the Ion AmpliSeq designer because I wanted to share with you a panel that these guys sell. It's a huge panel. It's 289 genes, nearly 300 genes, 16,000 primer sets, divided into 4 tubes. 4,000 primer sets. That's a boatload of primers in a tube looking at genes on this panel. So those big panels that are out there that we talked about today, Foundation Medicine, it's going to be very much similar to something like this. So 16,000 primer sets. Yes, well, we'll talk about the efficacy of those 16,000 primer sets perhaps over dinner. But anyway, that's the design of that product, and it seems to work. But if you look at some of the people who have investigated this panel, there is some issue around bias of the PCR. So some cases you can get 2,000, a depth of 2,000 for a particular target down to a 10x. So it does vary. And I talked a little bit about the rules of primer design. It's not always as easy as I put it up there. So the more complex it gets, the more difficult it is to keep things balanced so that you don't get a bias. But this Ion AmpliSeq has been tested by many academic labs who seem to like it. So here's the, I'm not going to go through every single one of these, so but here's basically a very top level view of product development that follows the route that you need to take if you're going to go through even partly to the validation step here, and further on if you're going to go into manufacturing and get some 510K clearance. The biggest step is this product requirement
You
document.
said 510K clearance?
Yeah. So it's 510 brackets, little k, close brackets, clearance. So product requirement document basically says, what is my product about? And you're going to lay out all the components, all the for want of a better word, the things that you want it to deliver on. Okay, so each one of those panels has gone through a product requirement document for you to know what it is you're going to build. And then some of the key things down here, we'll talk a bit about reagents, instruments. They can't just walk in off the road here. They have to be what's called IQ, OQ, PQ, installment quality, operational quality, performance quality. This is done as part of the regulatory compliance that labs need to follow. So you don't just take equipment in and start using it. It has to be tested and validated that it will deliver. And some other QC assays, very important. These tests are done with QC assays to check each one of the steps. You saw the process. It's quite an arduous set of steps. Again, not knowing what the processes are, but know that there are QC assays in place that test the quality of the product as it goes through. And that's very important when you have these discussions, especially with some of your customers who may not be in the know so much as to what goes on behind the closed doors of the lab. Quality, QC assay is very important. And then you go through a phase gate, what's called a phase gate, and if everything looks good, it's passed, and quality assurance is really the player there that lets it move to the next level. And then this is your feasibility, then you test it 3 times. You really want to see that you can build these kits, or these components that come together or these panels that come together to test for colorectal cancer, for example. You want to test to see if it works. That's what that's really working on. And then when you move to the validation step, is doing it again, but this time under stricter control. These are master batch records, which are called MBRs. This is done under draft, so you're still testing it out. You've got a little room to start playing just in case things don't work. But once when you get into that validation step, it's all locked down. And then you go through, test it 3 times. You've got all your parameters in place. Does it meet these criteria that you put in place? If it does, then you're ready to go. Here's the route. It's very long, very arduous, many steps. You've got to follow, you know, are you ISO 13485 certified? Do you follow the FDA QSR? There's a higher level of training required for the scientists. They're not working in a research lab anymore, guys, so you can mention that. These are highly trained people that are working in the lab, that are working in a regulatory environment. And so it's very important to stress that. I think there's confusion about scientists that work in a research and doing something like this versus really working in a regulatory environment. Okay, so these guys are trained, know what they're doing, they follow the clear recommendations, is traceability of documents, all the way through from the product coming in or the sample coming in through to being used and then the result. Okay, so but getting to the actual product itself, these number of steps which I somewhat alluded to in the previous slide. So one of the interesting things and important things to bear in mind is the reagents that come in to a clear lab. These are analyte specific reagents, ASRs. You'll hear perhaps the words bandied around. Wanted to bring it up because basically when those colorectal panels or the lung cancer panel or the neurosequence panel is put together, those components come out, they are higher grade quality components. They're brought into the lab and they make the assay. They do not make a kit. You cannot sell that kit or that assay. You can never do anything to it but use it, those components, in the lab to test for a sample that the request has come through for lung cancer. Okay, so these individual components come together, a bit like cooking, put into a pot and tested. You can never sell the components that are in the pot as a kit because you are not FDA cleared to do that. Okay, you're a lab developed test. That lab developed test remains in the clear lab. And why I bring up these reagents is because these analyte specific reagents are a high quality. Again, they will come in to you, but they will never be told, they'll never be on the sheet of paper that they can be used for X, Y, and Z. It'll just say KRAS primers. That's it. Okay? Again, because they're not supposed to be utilized for anything else. They're not supposed to be giving any medical use whatsoever. Just KRAS primers. How you use them is them following your standard operating procedures, SOPs. So the reason I bring it up is because that's very important. That stays in the lab. In vitro diagnostic tests, a bit different. You bring these high quality reagents in, you go through those steps, the verification, validation. The manufacturer takes that, those components and utilizes them and creates a kit that can be sold into a clear lab. As a clear lab that you folks have, you can buy kits and bring it in, but you cannot sell the products you've made, these tests, to anyone else. You can sell the patent, you can get the patent and give it to somebody, but they have to go through the same process that you went through to be able to utilize that test in their lab. Okay? So just bear in mind those distinguishing features. Lab developed tests, high level of regulation, FDA extremely high, and the components that come in, FDA can get put in a kit and sold. Lab developed tests stays in the lab, nowhere else. Yep, so as I mentioned, these are regulated by the FDA, subject to controls. So when the product comes in, these individual components, you have tests in place that test that they work. It's funny, right? Before you even get to use them on your test, you have to test that they work. So it's a QC assay, a quality control assay. That's very important to know. These QC assays are all in place to check everything is in place. Okay? So antibodies, specific receptor proteins, ligands, nucleic acid sequences, all basically binding, specific binding or chemical reactions with substances in a specimen. That's how they get in there, and they're intended for use in a diagnostic application or for the identification and quantification of an individual chemical substance or ligand, okay, which would be a piece of DNA with a mutation in it. As I mentioned, they cannot be labeled, these analyte-specific reagents cannot be labeled instructions for use or performance claim. They're just primers. FDA is very strict about this, but they are made at a very high level. So the manufacturer that you bought those primers from have these incredibly high levels of quality control too. So there's documentation all the way through, and that documentation is brought into your CLIA lab so that when you get audited, you can show from the very moment that that product, any of those individuals' components come in, that they are there, they've been qualified, tested, and they work. Manufacturing is the next step. If you're going to create a product that's going to go out to the market, you can either do the in-house, have a manufacturing suite, or use a contract manufacturing organization, and all these components are what go into testing and validating that it works. So pre-formulation, formulation development, stability studies, that's another important thing. The components have to go through stability studies, because if they're going to be in a kit and they're going to go out on someone's shelf, they're going to be in a freezer, will it break down? So when you have a shelf life on there, it doesn't matter even if it's 1 second past midnight of that day, you can't use it. So stability studies are important, and then you've got clinical trials and so forth. Scale up is very important as you get bigger and better. This is for FDA. And then commercialization, you've got CE marking, which I'm sure you've seen on many of your packages and instruments. CE, it's a French word, Certificate de Europe. And the FDA 510K, that's for selling your product in the US. And then other countries may require you register so that you can sell your products, okay? So big difference then with the lab developed tests. They only go so far. They're very much focused in your lab compared to whether you want to sell this in the US as a kit that can go out to any other lab, a CLIA lab in particular. So here's some of the differences. The LDTs are performed in a CLIA lab, which is Clinical Laboratory Improvement Amendment. I'll talk a little bit about how CLIA came about. Again, it came about because the government wanted to ensure that there was some regulation over these products that were being built, as there was a variety of standards, so standardize everything. This is not a single standardized test, of course, and it's comprised of ASRs, which we talked about, that come together to make the assay, okay? And as I mentioned, you can't sell it as a kit. For EU, if you want to sell your product in Europe, you have to use a regulated, they're used in regulated labs, validated on retrospective samples, and manufactured under CGMP, and contains an instruction for use. In a CLIA lab, the instruction for use is your standard operating procedure. If you're going to sell a product, you have to put the instructions in there, and they have to be very, very, very carefully written and easy to follow. And then for the FDA cleared product, somewhat similar, except that there needs to be a clinical study. These are all performed in some kind of CLIA lab, hospital laboratories, and manufactured again as a kit. This was very interesting that I wanted to put together was LDTs and FDA cleared, as in what they're used for. So, as I've mentioned, you can only use these LDTs in a CLIA lab that designed it. Okay, that's it. Your lab is the only place in the world that can use these panels. No other lab can do it. They can take something similar and they have to go through all the processes that your lab went through, but they cannot use this panel. You cannot sell it. Okay, you can, like I said, you can, if you have a patent around these, you can sell the patents, and then everybody has to go through the same process. But it's your lab is the only place in the world that does these tests. So all samples that come into your lab that wants this lung cancer panel done, it's the uniquest place in the world for that. People can copy it, but this is yours. Okay. For the FDA then, once the assay is cleared, it can be used in any CLIA laboratory, has to be in a CLIA lab. You purchase it from the manufacturer, you follow those instructions, and then, of course, the price is a lot higher. Okay, so let's change gears a little bit and talk about those laboratory services that provide the tests. There are several of them on here, and that David and I went through this. We thought it'd be good to just take you through anything from the CLIA lab all the way down to somebody in their own home that can either do some of these tests or send out the test to a lab. Okay, so you've got the hospital labs, of course, the reference labs, public health labs, point of care, POC, I'm sure you've heard that acronym. So that's basically in the doctor's place and they do the test there and then direct to consumer. So I'm going to walk through each one of these, give you a good sense as to what these guys can and can't do. So let's start off with Clear Lab. What does it mean? How to get it put into place? As I said, it was an opportunity for Congress to really cement a level of standards that all labs must meet. So in 1988 they did that, and they came up with the Clinical Laboratory Improvement Amendments Act. So it's established quality standards for all non-research labs, specimens derived from humans, and the purpose is really diagnosis, prevention, treatment, impairment of or assessment of health. Anything that relates to you as a person that's going to tell you what could be or could not be wrong with you. Okay? It requires that you get certified by the DHHS, Department of Health and Human Services, and as David was telling me, the Centers for Medicare and Medicaid Services, CMS, administer all the clear laboratory certification for the DHSS. Okay? That's big. Just knowing that's important. And in conjunction with the FDA and the Centers for Disease Control and Prevention. So it's all done together. These are all tied up. But this was a good way for the FDA to say, all right, if we can't bring these standards in, then let's bring these standards in and make sure that it's standardized across the board. But like I said, it's still a issue of contention with companies who want to make money as to how much the influence of the FDA is over the clear labs and trying to drive up the standards even higher. Standards then really bring in accuracy, reliability and timeliness of patient test results so that you don't sit there and say, oh, I don't know, maybe a couple of weeks, maybe a couple of months, right? So the aim here is that you say 3 days, you deliver on 3 days, and everything is documented so that when you get accredited again, you get audited, you'll show the documentation that there is a correct turnaround time. You're not suddenly saying, oh, it's 3 days for one test and it's 10 weeks for another. Standards are national and are not Medicare exclusive. It applies to all providers rendering clinical laboratory, and regulations are based on the complexity. This is kind of interesting because if it's a simple test, it doesn't have too much of a documentation or auditing versus moderate and high complex. So those are very important. But again, I put in red here, it's about documentation, it's about quality, and it's about quality assurance. That means basically when that patient sample leaves the clinician's office, it is tracked from that point onwards through your system and back out to the clinician with the result. Okay? And if it's not, the audit will catch it. And then you'll be held suspended until you fix some of the issues. There are 5 types of laboratory certification. Yours is the most complex, and we'll get to that. The certificate of waiver is the simplest tests. It just permits a lab to perform some simple blood tests. Then there's one that's performed for microscopy that some people, clinicians like to do. Dentists perform them as no other test than certain microscope procedures. Then there's certificate registration. This allows the laboratory to conduct moderate or high complexity testing. Yours is probably high complexity, compliant with all regulatory requirements. And then there's a biannual review, which is a COC. Basically, this is the auditing point. So observation of the laboratory's past and current practices are looked at, documents are brought out based on the auditor who wants to sit there and see it. There's interviews with the laboratory's personnel and review of the relevant documents, as I mentioned. So having been in an audit myself, not an FDA audit, but audits to get certification, it's pretty rigorous. It can be 2 days, and literally they say, right, let me see this documentation, and then you run out and you get it, you bring it in, and the person sits there, and you sit there waiting quietly, and then the person says, so tell me then, how did you do this? And that's how it begins, so forth. So, certificate of accreditation, which is what you have. There are 6 basically non-profit accreditation programs approved by the CMS. One of them is the CAP, College of American Pathologists, and I believe these guys have accredited you. and it's designed, these guys really go out of their way to make sure that you not only meet the clear regulations, but if you can, exceed them, okay? And they're an independent body, you can't bribe them, you can't do anything with them. These guys come in, and the people that do this job do the job very well. They enjoy their job. So the CMS has granted basically these type of non-profit, non-profit organizations, the the accreditation program deeming authority. That's what I wanted to mention. So CAP can basically say, you have lost your accreditation because you don't meet these standards, you're shut down until you do. That's how much power they have. So that's important for the CMS to know that these guys will do that, and that they have the power to do that. And then, of course, there's also some joint commission to meet many state certification requirements, which I believe you guys are trying to get into, New York being one of the most difficult, of course. More reading if you're interested about clear labs. If it tickles your ivories, it's kind of detailed information, but good stuff to learn a little bit more. Hospital laboratory testing. So let's go through and talk about then what all these places have. Hospitals will have clear labs, okay? And they tend to be of a size that is really proportional to the community. They do state testing, volume testing, stat testing, which immediate, not state testing, volume testing, and they're used by inpatients as well as outpatients. They can typically segregate into microbiology, hematology, chemistry, and blood banking, and they are specialized, can be specialized too, with genetics in there as well. So a reference laboratory, somewhat different. It's a private enterprise. They perform sort of low but specialized tests. They require the specialized equipment that go with that, and these type of genetic tests fall under that. So they can be in reference, performed in reference labs. Tests that are referred to these are from physicians' offices, hospitals, maybe overspill because they can't manage the amount that's going on, so they'll send them out, and other healthcare facilities. And they can be some distance away from the hospital or the doctor. Okay, so it's not just like round the corner. So that's where the whole tracking mechanism comes in. Public health laboratories are somewhat slightly different. They're run by state and local health departments. They're usually focused around infectious diseases and environmental hazards. So if you're hearing about public labs, they tend to be the ones that will monitor flu progress during the flu season. For example, you'll get updates on the television, CNN and that's coming from these public health labs. So they usually keep an eye on the prevalence of certain diseases, as I mentioned, flu, and if there's any outbreak, that big E. coli one a few years ago in Germany, those would be kind of public health laboratories that would manage that if it ever happened it does happen over here. I think there was a spinach outbreak just recently with E. coli, right. So they investigate, screen newborns for various genetic diseases as well, and specialized molecular tests. They can offer those too. But typically it's a go-to lab for first responders in situations that can occur. And in particular, the CDC, FBI, and other federal agencies come together. They form this laboratory response network, which is very important for them. It allows information to get fed in, testing to be done, and then passed out, especially around biochemical and biological terrorism and public health emergencies such as flu, or if there's an outbreak of E. coli in food, etc. Point of care is big. As you know, it's a multi-billion business. It's going to be, numbers are thrown out by 2020, it'll be like $10–15 billion. This is basically the opportunity for you to walk into your doctor and get your testing done there and then, and the doctor is able to say, right, here's what you need to take on, here's what you need to do. So point of care is very important because this eventually and ultimately goes down that route. There's no doubt about it. It has that potential that one day a PGM could sit in a doctor's office and your blood's drawn or samples taken and it's just put into the machine, and maybe 24 hours later you're going to get a result. But basically these do occur in offices, doctors' offices and clinics, Hospitals perform specific tests at the patient's bedside. So that's another place where point of care occurs. And if you've been into hospitals recently, somebody who's very ill, you see a lot of instrumentation around them, and it's basically that's the kind of point of care. They're doing quick tests right there and on the fly, and the nurses are taking measurements every so often and recording it, and especially in the intensive care unit. So labs within physician offices tend to be very small, menus are limited, but I'm sure as the equipment gets better, smaller, easier to use, that's going to change, but it's expensive. It requires personnel that needs training, and so of course the samples that can go through the equipment then is also small, and the results, rapid results, and convenience is what comes out of that, and that's the promise of point of care. So typically then you got blood glucose tests, blood gas monitoring, whole blood analyzers for cardiac markers. These are the kind of things that sit by your bedside, give you quick results, can actually make or break a big decision when a doctor wants to know what's going on with the patient who could be going through some difficulties. So powerful stuff as that grows. And then the final one, we've heard a lot about these things. Direct-to-consumer genetic testing is one of the biggest growers in this industry. It's powerful stuff. Quick swab, stick it in a tube, off it goes. 23andMe, for example, will tell you what mutations you have in your genome. That's pretty much it. But then, of course, the big concern is what does it all mean? And how do you take that to your doctor? And how does the doctor get to understand how to apply it back to you? So some of the simpler ones, such as pregnancy tests, glucose monitors, and a fecal occult blood tests for diabetes and colorectal respectively, as well as prothrombin time testing. These are the simple ones that are cheap, easy to use, disposable, and make for comfort by just buying it and doing it at home. As I said, the samples typically can be collected, mailed, then you get a result. Hepatitis C tests and HIV antibody tests are something that easy to do that way, but there may be a prescription required to get that test from the over-the-counter. And then, of course, the convenience, the privacy, the rapid results is the excitement of the direct-to-consumer tests here. And like I mentioned, a buccal swab straight off to wherever, and you can come back with all sorts of information, BRCA1 or 2, if you want to test that, and so forth. And that's really it. It's a short one, but it gives you a good sense. Tomorrow, if you want to keep going, no. So tomorrow then we'll talk about competition. But today, that's really it. So I hope at the end of this module 3, you get a good sense as to what the CLIA lab is and what these tests are doing in there, and that they are under regulatory compliance, and that they are follow strict documentation with quality assurance second to none. Okay, so that's very important, especially if some of the physicians don't understand what CLIA labs mean, and the fact that it has to go to a lab. Why isn't it going to a hospital lab? What's this lab all about? This is very important to be able to communicate that simply and effectively to them, so that some of these clinicians understand that. Like I said, some of the second-tier hospitals that don't quite have that full knowledge don't always understand what's going on, especially genetic tests. Genetic tests in a CLIA lab? Really? I didn't realize. Why? So forth. So just some of those key points there, and hopefully give you a good sense as to how that landscape lies. That's it.